test. So Ames test is done to test the potential of any chemical that it can cause mutation. So to test any chemical that it has a potential that it is a potential mutagen. So mutagen are those which cause mutation. So if we want to test any chemical that it can be a mutagenic agent, that it is a mutagenic agent or not, we perform generally Ames test. Okay, so the Ames test or Salmonella typhimurium reverse mutation assay, we will discuss about this, and we will also discuss about that how people have combined this assay along with mammalian mammalian liver extract, to see or to further confirm or to evaluate that of chemical is a mutagen or not. Mutations are sudden permanent change in sequence of DNA. So what are the mutations? Like if A pairs to T, then now instead of A there is G or there is C. So there is a change in base pair potential. So when there is a change in base pair potential, we say that a mutation has occurred. Okay. So mutations are any sudden permanent change in sequence of DNA. Okay. And who gives mutations or who produces mutation are mutagens. Mutagens are those that causes mutation. Mutagens are of various types. They can be chemical agent, they can be physical agent, there are biological agents like viruses who has tendency or who they have potential to cause the sudden permanent change in our DNA. To test the potential of any chemical that it can be a mutagen or not, we perform a standard assay called Ames test. What is Ames test? Ames test is a test to determine the mutagenic activity of chemicals by observing whether they cause mutation or not. And what will be the parameter on which we will be assessing that whether they cause mutation or not? It is basically this is held by bacteria Salmonella, where we do bacterial reverse mutation assay. We'll look into this. So what is the principle of this Ames test? Several strains of bacterium Salmonella typhimurium carry mutation in genes involved in histidine synthesis. Okay, so there are genes which, let's suppose, I guess you have heard about lac operon or tryptophan operon. Similarly, there is a histidine operon in case of Salmonella typhimurium, in which there are various places in the structural region or in the structural region of histidine operon or his operon, which get mutates, because of which there is no histidine biosynthesis in these strains. So there are several strains of bacteria which of Salmonella typhimurium which carry mutation in genes involved in histidine synthesis. So I am talking about these strains. These strains, all Salmonella typhimurium, they have histidine operon, and they code for various proteins which help, or various enzymes which help in histidine biosynthesis. Let's suppose there is a mutation in structural region of this histidine operon, because of which now histidine biosynthesis is not there. So these Salmonella typhimurium will be called as histidine negative, or those Salmonella typhimurium which are not capable of making their own histidine because of the mutation in histidine operon or the structural region of the operon. And I guess you can correlate this phenomena very well with the lac operon. These strains are histidine negative auxotrophic mutants. We know that auxotrophics are those organisms which are not capable, or which are not capable of making a particular chemical, and they depend upon external source of that chemical to survive or to grow. So these strains are histidine negative auxotrophic mutant, or the deficiency mutant, they cannot produce histidine and thus require histidine to be supplemented in culture medium for their growth. So what I am so so what I am trying to explain is that that in Salmonella typhimurium, in Salmonella typhimurium, there are various strains which are auxotropic for histidine, because because there are mutation in histidine operon or in the enzymes which help in the histidine biosynthesis, because of which they are not capable of synthesizing their own histidine and they are dependent on external source of histidine for their growth. So if we can convert these Salmonella typhimurium mutants, which so if we can reward the mutation, so if we can reward those mutations which are not, which are causing these strain to be histidine negative. So let's suppose there is a mutation in histidine operon, and in the structural region where a stop codon has came. Now, if I can revert a mutation in a way that this stop codon is now no more a stop codon through a substitution mutation, that now it can code for some particular amino acid, so that a protein, a full-fledged polypeptide can be formed